Welcome, everyone. My name is Wala Muhammad, Market Segment Manager for Complex Disease at Oxford Nanopore Technologies. And it's my pleasure to welcome you all to our Knowledge Exchange webinar today. I'm delighted to be joined by Ashan Musafir and Patrick Murphy. Thank you for joining us for this online session, where we will be looking at decoding complex disease through multiomics. After the talks are over, the team will be available to take your questions in a live Q&A. So please add your questions into the Q&A tab, which you can find it to the right of your screen. You can also upvote the questions you would most like answered, and we will try to get through as many as we can. This webinar is being recorded and will be made available on demand after the broadcast. Well, complex disease are rarely explained by a single variant or one biological layer. GWAS has been extremely valuable for undefined regions of the genome associated with disease risk. However, in many cases, it tells us where the signal is without fully explaining which variant is responsible or how it affects biology or whether it's truly driving the disease. Complex disease also is highly polygenic and context-dependent. Their biology may involve regulatory variation, structural variants, different transcript isoform, and epigenetic changes. Some of this information can remain unresolved when we rely only on SMP level association or short-read data. So we usually begin with GWAS, which undefines the genomic region associated with the disease risk. And then the next step is fine mapping, where we try and narrow that association to the most likely causal variants or gene. Then undefining a candidate is not enough. We then need to understand the biological mechanism. How does the variant affect the gene regulation, transcript structure, expression, or another functional pathway? Finally, these findings help us build evidence for causality and determine whether the observed change is genuinely contributing to the disease biology rather than simply being associated with it. So the key message here is that the next step in complex disease research is not simply finding the risk loci, it's connecting those loci to biological function and explaining how they drive disease. To understand complex disease, we need to look beyond a single tab of biological information. So the advantage of nanopore technology is that the same core sensing platform can provide access to several complementary layers of biology. At the DNA level, long-read sequencing can reveal single nucleotide variants, structure variants, repeats, complex genomics regions, and also haplotype information. And because native DNA can be sequenced directly, methylation and other DNA modification can also be detected alongside the sequence without requiring a separate bisulfite conversion workflow. So at the RNA level, nanopore sequencing can capture full-length transcripts, helping researchers distinguish between different isoforms rather than relying on one gene-level expression. And direct RNA sequencing also can be sequenced directly, so it preserves the information from the native RNA molecules, which will support the study of RNA modifications and epitranscriptomics. The real value here, it comes when these layers are integrated. So instead of studying DNA variants or RNA, you can connect all of them to build a more complete model for disease biology. So the key message here is one underlying technology can provide progressively richer biological insight from sequence to regulations to function. Also, the platform is designed to support different study sizes, sample types, and experimental requirements. Oxford Nanopore offers flexible library preparation option to match experimental design, from rapid to standard, long-read sequencing to ultra-long DNA, and also rapid PCR for raw input workflow. Direct RNA and cDNA sequencing also, it's flexible, and you can hear more about it in Patrick's presentation later. The platform can scale from small pilot studies to large cohort. And for multiomics project, the Prometheum 24 here can provide suitable scalability depending on the required throughput. Finally, our epigenetic analysis software provides intuitive workflow to support data analysis and interpretations. That flexibility is really important because the ultimate objective is not simply generate more data, is to turn those data into biological understanding and useful biomarkers. This brings all these elements together. So the pathway moves from discovery to mechanism, biomarkers, and translation. The journey begins with the discovery science. Long read and native molecule sequencing can reveal biology that may remain hidden to conventional methods, including structure variation, complex genomics region, alternative RNA isoforms, and methylation pattern. The next step is understanding mechanism. DNA can identify the risk variants and provide haplotype context. RNA can show the downstream functional consequencing through changes in expression or transcript structure. Also here, methylations add the regulatory context and may help explain why a genetic effect is active in a particular tissue or cell types or disease state. So when all these layers are combined, researchers can begin developing multiomics biomarkers based on integrated DNA, RNA, and methylation signature. This signature may provide more biological information than a biomarker based on one data type alone and can support improved disease stratifications or cohort selection. The final stage is translations because the same platform can scale across different devices, as I showed you in the previous slides, and throughput requirements. Workflows developed across discovery can potentially be expanded into larger studies and translational research programs. So the value here and the value pathway is clear. We move from discovering previously unresolved biology to understanding disease mechanism to developing multi-omic biomarkers and ultimately towards translation. With that introduction to the complex disease challenges and the role of multiomics, I will now hand you over to Ashan, Senior Field Application Scientist, who will take us into scientific evidence and application in more detail. Welcome, Ashan, and over to you.
Hi, everyone. My name is Ashan Musafir, and I am a Senior Field Application Scientist at Oxford Nanopore Technologies. So in this section of the talk, I will be walking you through a few recent articles that highlights the importance of, a multiomic view of the genome when studying complex disorders. So the first question, why do long reads matter? For over 2 decades, since the Human Genome Project, scientists have been working with a draft of the Human, Book of Life. And as you know, it's fraught with missing chapters and blank pages. But recently, genomics has entered a T2T era, from telomere to telomere. So this also means that mapping a chromosome completely from one end to the other without a single gap is possible. But is there a problem there? Now, getting a T2T genome has sometimes been a luxury. You need maybe multiple platform data, or you also might need high computational power. However, that's until now. Developed by Dr. Haiyue Sheng, an assistant professor at Yale University, enters high-FISM ONT. And this is a new brilliant algorithm. So instead of looking at letters or nucleotides in isolation, high-FISM ONT uses a mathematical framework called joint phasing. So what that realizes is that true genetic variants are team players. They always line up in consistent phase, with other nearby variants, inheritance from both parents. But recurrent sequencing errors, on the other hand, these are loners. So they don't cleanly align with surrounding variants. So by mapping these using networks of advanced dynamic programs, the algorithms have the ability to filter out these systematic glitches. So what that does is, it corrects these standard reads until they are virtually error-free. So as you can see on the table, high-FISM ONT not only reconstructs more complete chromosomes from the number of scaffolds and contigs, that you can see here. But using the exact same standard data, it also significantly decreases the computational demands, as you can see from the wall time in hours. But how does this capability play a role in the understanding of complex disorders? Now, in clinical research, sometimes de novo assembly is needed to reconstruct these medically relevant genes. And these are those genes that are often difficult to resolve using conventional alignment-based approaches. So in reality, the true power of this publication is not just computational, it's actually practical. And thanks to high-FISM ONT, the ultimate goal of genomics, having a complete gapless map of your DNA is now standard practice. So now from this, we know that long reads can resolve these complex regions. But remember, long nanopore reads also have native sequences. So this is native sequencing, which provides methylation data. So let's now look at how you can add methylation and how you can study methylation and its role in complex diseases. As many of you know, the ApoE gene encodes for a lipid transport protein, which is known as apolipoprotein E. Now, this gene has three major alleles, and these three alleles are defined by two SNPs or single nucleotide polymorphisms in exon 4. So the E2 allele is neuroprotective, the E3 allele is neutral, and the E4 allele increases the risk of late onset Alzheimer's disease. However, the genetic sequence alone doesn't tell the whole story. Now, this next preprint that I'm going to talk about from bioRxiv bridges that gap between genetics and epigenetics by looking at how you can use DNA methylation, and that differs between the maternal and paternal chromosomes, where you can do haplotype resolved sequencing. Now, past studies have tried to link methylation, and especially when it comes to this ApoE locus and its relationship to Alzheimer's risk, but they do face a major hurdle, and that major hurdle is bulk sequencing. So now, if you think for a moment of traditional methods of sequencing, if you are heterozygous, let's say you carry one E3 allele and the other E4, in bulk sequencing, the signals get blended. So bulk sequencing gives you the average that masks whether the epigenetic change or the methylation is specifically on either the high-risk allele or the neutral allele. So the work done by Dr. Riley Jenner at the Center of Alzheimer's and Related Dementias at the National Institute of Health sets out to solve this blind spot that we have. So in this study, the authors had three major objectives. The first objective was to map the DNA methylation with haplotype resolution across this ApoE locus. The second objective was to determine if these allele-specific signatures are consistent across different tissues. And in this case, specifically looking at human postmortem brain tissue, as well as peripheral blood. And finally, the third objective of the study was to assess whether these epigenetic variations correlate with actual clinical Alzheimer's disease, the presentation and its pathology. So to achieve this, Oxford Nanopore long reads were used to track those individual SNPs and face the DNA. And this would also mean they could confidently assign every single CPG site to either the maternal or the paternal chromosome. So what this group did is they looked at a publicly available dataset of 300 plus postmortem frontal cortex samples from two different cohorts. So the first cohort was called NABEC, or the North American Brain Expression Consortium. And within this sample cohort, there were a couple of samples with European ancestry, and a second cohort known as HBCC, or the Human Brain Co-Collection. One thing interesting here was they also had some array-based methylation data from another study called the ROSEMAP study, which had participants from a European ancestry. Now, something very interesting here is they correlated the data from the ROSEMAP study to this particular cohort, which is the NABEC cohort. And if you look at the 46 CPG sites that are shown on your graph over here, you can see there is clear correlation between the data coming from the array as well as the nanopore long reads. So that gives you confidence where you have two methods, two orthogonal methods giving you the same result. And if you really look closer into the correlation, we have a Pearson R score of about 0.98. However, if you dive a bit deeper into the data, there is a clear difference when you use, an array-based technique versus the nanopore technique. Now, what you do get from Oxford Nanopore data is more resolution, because remember, with native sequencing, you are able to get all the CPG sites within that region. So if you compare this ApoE gene cluster that we're looking at in this picture here, what you see at the top is 46 sites that have been evaluated using the array, whereas at the bottom, you see the exact same region, but now you're looking at 1,556 different CPG sites. So you're getting more resolution. Let's dive into the results. One of the major findings is that DNA methylation at this ApoE locus is highly allele specific. So when you look at an heterozygous individual, for example, the E3 and the E4 alleles, the researchers found that there is a stark difference in the epigenetic landscape of the E3 chromosome compared to the E4 chromosome, even though it's the same cell environment. So the E4 haplotype exhibits distinct hyper or hypomethylated regions, and these regions are particular around the promoter and the enhancer elements that regulate their apolipoprotein expression. So this proves that genetic variation at this locus is directly dictates its epigenetic state as well. So the next is that they also compared the brain tissue to peripheral blood. And interestingly, they found that while some allele-specific epigenetic markers were unique to the brain, a proportion of them haplotype-specific signatures were also conserved in the blood. And this is a crucial finding because this suggests that peripheral blood could potentially mirror the epigenetic status of this ApoE locus in the brain, and that opens the door for minimally invasive biomarker development. And perhaps the most clinically relevant finding is how these signatures correlate to disease. Now, the authors observed that epigenetic variations on the E4 haplotype were significantly associated, as mentioned before, to an increased Alzheimer's pathology. So, such as higher amyloid beta plaque burden and faster cognitive decline. So this association is much stronger when you look at phased haplotype specific data when you compare to a traditional unphased bulk methylation data. So what is the underlying mechanism? The authors suggest that these allele-specific methylation changes alter the binding affinity of some of the key transcription factors. So when these E4 alleles undergo these epigenetic shifts, it's likely to dysregulate the ApoE expression. And because ApoE plays a critical role with the lipid transport, the amyloid beta clearance in the brain, even a subtle shift in an allele-specific expression can compound over time, and that would then accelerate the neurodegeneration. So why does this publication matter? It fundamentally changes how we view genetic risk when it comes to Alzheimer's. It's not just about having the E4 gene, it's also about the epigenetic state of these specific alleles. So moving forward, this kind of opens up an exciting avenue when it comes to precision medicine. We could use blood-based epigenetic screening to predict disease onset. And that further highlights that the ApoE locus is the potential target for epigenetic editing and therapeutics. So these findings together brings us one step closer to understanding the complex interplay between the DNA sequence and our epigenome when it comes to neurodegenerative diseases. So we know that long nanopore reads not just help us assemble the genomes, but these native reads also provide us a glimpse of its epigenome. And that is true value. So let's now add another layer into this. What about looking at transcriptomics? So traditional RNA-seq experiments like to study or measure the gene abundance. But the question is, does that tell you the whole story? But let's take a step back. Now, similar to native DNA sequencing that I explained before, it is possible to do native RNA sequencing with Oxford Nanopore technologies. So what happens is these intact, unamplified RNA strands are translocated through the pores. And as they translocate through the pores, we create the squiggles get base called, and these base call data also captures all the different modifications that are present in an RNA strand. And as you know, RNA has many more modifications if you compare that to DNA. Not only that, when you do direct RNA sequencing, you also have the ability to estimate the poly A tail lengths simultaneously. So to understand how the study of epitranscriptomics adds as an additional functional layer, let's have a look at the next publication, which goes into more depth of the use of epitranscriptomics, and that then adds another layer into the study of complex diseases. Now, in this particular paper, we've known for years that the human brain possesses an incredibly complex transcriptome. And this transcriptome is driven heavily by alternative splicing. Now, we also know that chemical modifications on RNA, the epitranscriptome, plays a part when it comes to the regulation, the memory and neurodegenerative diseases. But now, how do we connect the dots? How do we connect these two worlds has always been another technical hurdle in traditional methodologies. So the simple question that comes to mind in this particular publication is, now genes produce multiple RNAs, which are isoforms and transcripts. Now, different isoforms sometimes have distinct or opposing functions when it comes to cellular development. But which of these isoforms are regulated by a modification? In this case, let's say M6A modifications. And how does that dictate its fate? So let's dive deeper into this publication that was published recently in Science Advances, titled Isoform Level Profiling of M6A Epitranscriptomic Signatures in the Human Brain. In this study, the authors examined postmortem brain samples from donors without neurological disorders. So these are normal, patients. They focused on three distinct functional regions of the brain: the prefrontal cortex, the caudate nucleus, and the cerebellum. The good thing is they also maintain rigorous quality control. If you look at the data, where the samples with an RNA integrity greater than 7 were actually sequenced. So what that means is you have these full-length RNA molecules that can give you, more resolution to look at different isoforms. And their data set was massive. So they generated over 52 million high-quality reads and effectively took those reads and mapped it against 22,000 genes and 62,000 distinct transcript isoforms. Now, what they were able to do with this is, firstly, the authors built the first comprehensive single nucleotide isoform level atlas of this M6A in the human brain. As I mentioned, there were over 9,000 differentially expressed genes and over 16,000 differentially expressed isoforms between the different parts of the brain or the different regions that they studied, as you can see from these graphs over here. Now, here's an interesting finding. Now, when they studied these transcripts, as of course, within a certain gene, there are multiple isoforms. And in this case, if you look at the SYNPR gene, there are 4 different isoforms. And the SYNPR encodes for a synaptic vesicle component. It's also known as synaptoporin. And this was expressed in all 3 regions of the brain. However, if you look at it a little bit more deeper, you will notice that one particular isoform over here was only expressed in the cerebellum. So if you look at the prefrontal cortex and the caudate nucleus, it contains the different isoforms, but the cerebellum is just one particular type. So if there's only one isoform contributing to the expression, it will be interesting to see what governs this or what regulates this particular isoform. Moreover, this study also discovered more than half of the total detected isoforms, or 55% to be exact, were also modified. And when it comes to these modifications, there were 57,000 M6A sites that were identified. The median modification rate was about 0.66. What that means is if you take a single isoform type, roughly 2/3 of those molecules carry a modification. That also kind of highlights just how widespread and tightly regulated these epitranscriptomic markers are. However, one of the most compelling insights from this paper is the spatial patterning of these modifications. These authors discovered that there is a brain region-specific regulation in particular the prefrontal cortex. The prefrontal cortex exhibited a highly distinctive profile. So not only did it showcase, a unique set of modified isoforms, but it also contained the highest proportion of previously unannotated M6A sites. So what this implies is that complex cognitive processing is unique to the prefrontal cortex, might be driven by, a context-specific epitranscriptomic event. Lastly, they also identified a distinct population of transcripts, which they termed hypermodified isoforms. And these specific transcripts are densely methylated across all the studied brain regions, and fundamentally around the excitatory neurons. And this also suggests that M6A tagging might be a dynamic process and might also provide the baseline architecture to preserve some of the neuronal biology. So what are the biological implications? By proving that M6A modifications are significant between these different isoforms of the same gene in a region-specific manner, this study really changes how we interpret gene regulation, right? So dysregulation of M6A has always been implicated in neurodegenerative and neuropsychiatric disorders, but this atlas provides the coordinates. It provides those exact coordinates that are required to study disrupted splicing or aberrant RNA methylation work. And this together can drive these disease phenotypes. So to conclude, the first study showed the importance of how long reads assemble these medically relevant genes in an economical manner. The second study showcased the importance of haplotype phasing and studying allele specific methylation and its role in gene regulation. And the final publication added the layer of epitranscriptomics to better understand the mechanisms that govern biological processing. So I hope with these publications you get a glimpse of how Oxford Nanopore technologies have the ability to look at a completely multiomic view when it comes to studying complex disorders. Thank you for listening, and let me now hand over to Patrick to go over some of our newly released RNA barcoding kits and some of our enhanced cDNA protocols. Thank you.
Thank you, Ashan. We now go to Patrick Murphy, Market Segment Manager for Single Cell. Over to you, Patrick.
Great. Thank you, Walla. So now I'm going to pivot us from DNA over to RNA. And I really like studying and understanding transcriptomics because it adds a really nice dynamic layer to our understanding of biology and disease. And that's because RNA reflects, of course, the active state of the genome. It tells us which part of the genome are being used, in which cell types, and under which biological conditions. And so this is really important, especially important for complex disease, where the causal biology might not exactly be explained by the DNA sequence alone. The same genome, of course, can produce very different cellular states, and that can depend on tissue type, the environment, the disease state, kind of just where those cells are in the body and what's going on with them. Even if they have the same DNA, they could have different RNA profiles. And so traditional short-read RNA-seq, it's a very powerful method, of course, really great at quantifying gene expression, but short reads often provide only a really fragmented view of the transcripts themselves. They don't provide great isoform resolution, novel transcript discovery, which can make it really difficult for us to know which full-length isoforms are present in our sample. Oxford Nanopore sequencing adds a lot of additional value because our long read method will provide transcript reads that can span much of the transcript, often from end to end. And this means that we can get, obviously, full-length isoform resolution, as well as, since we've got that full-length transcript, the ability to do novel transcript discovery. That lets us then go from just asking, how much of a gene is present, to then asking questions like which actual isoform is expressed, how is it spliced, is there a fusion, what's going on with the biology here. And on top of that, Oxford Nanopore also has an additional solution that gives us transcriptomic information, and that's our direct RNA nanopore sequencing approach. And this will actually sequence native RNA. So in addition to getting the quantification and the transcript aspects that we would get from long-read cDNA sequencing, we're also going to be able to get information about the RNA molecule itself directly. So we can get RNA modifications that are present there through the direct detection of sequencing. And we're also going to be able to sidestep potential biases from RT and PCR because we're, again, sequencing that RNA transcript directly. So going back to that property of Oxford Nanopore sequencing, that it provides transcript level information, one of the really big reasons to, sort of be interested in and care about transcript level resolution is alternative splicing, right? So a single gene can actually produce multiple different RNA transcripts through the process of alternative splicing. And these different transcripts then, through processing, can then lead to different protein products or just generally different overall behavior of the molecule. So the gene's really not always the right unit of measurement that we should go for. Two samples might have very similar total expression of a gene, for example, but they can have very different isoform compositions. And for example, one isoform might be enriched in the disease, other might be enriched in the healthy tissue. One might encode a functional protein that's tethered to the membrane, for example. Another might produce a non-functional protein that's soluble. And so, this could be a situation that might happen in cancer, for example, where you get sort of like decoy proteins, for example, that could be soluble and sop up immune receptors or things like that. With short reads, we can often infer sort of what's happening with the alternative splicing from the fragments, but with long read Oxford Nanopore reads, we can observe much more of the transcript directly. We can see the whole thing or much of it across the full molecule. And this gives researchers a clearer view of the biology that's going to sit between the genome that we were talking about, as well as the proteome. The scale of isoform diversity is really quite substantial. Around 95% of human multi-exon genes undergo alternative splicing, and alternative splicing is associated with quite a few different disease states. On average, each human gene is going to have multiple isoforms averaging around 6 or slightly more than 6. So there's a lot of additional information there that we can start to understand when we view things from the transcript lens rather than just at the gene lens. And there's still a lot to be discovered as well. One study recently found that about 80% of the isoforms that they were identifying with long read were novel. So there's still a lot out there. And for complex disease researchers, this has some major implications. Just the general, the isoforms themselves have some major implications, because from studying our biology, if we collapse everything down to the gene level, we may be averaging together biologically distinct transcripts and potentially missing out on disease relevant signals. And so I think this Alzheimer's disease example really makes this point quite clear. What the authors saw here was that almost 100 different genes in their study, when they looked at the gene level and compared between healthy and diseased, they saw no differential expression in these genes. But what they went on to do further was look at the transcript level, and they could see the individual isoforms from the gene now show a very clear disease-specific expression pattern, right? So in this example, differential transcript expression revealed patterns that were not visible when all the isoforms were collapsed into this single gene level measurement seen here on the left. And so these signals would have been missed if the analysis had just been stopped at gene expression. Here's an example as well of one specific transcript, or one specific gene, I should say. So TNF superfamily member 12. You can see here on the left, not differentially expressed at the gene level, but we can see when we look at these two different isoforms of this gene, very different relative expressions, let's say. So in Alzheimer's disease, 219 transcript is high. It's down in Alzheimer's disease patients and up in controls. And so we see very different expression patterns. And then ultimately, if you look at the gene level, these look like they cancel out and like there's no difference, but really there's an isoform switch that's occurred. And so to that end, Oxford Nanopore is really interested in getting and making sure we have the longest transcript length so that we can get you information about the transcripts that are present in your sample. And so I'm happy to share that just last month, we were able to launch a new protocol that gives significant length increases from our cDNA bulk transcriptomics protocol. And the key performance measurements here can be seen on the right. And this is a length distribution plot where we're comparing the previous protocol in gray and the new protocol in blue. The updated protocol, we can see shifts towards much longer reads. So the median read length increases to around 1,700 nucleotides, and the N50 increases to around 2,500 nucleotides. And this is quite important because longer reads are going to improve our ability to span transcript structures. It's going to improve our ability to resolve isoforms and capture more complete molecules. So this update strengthens the value prop, I would say, of long read sequencing as well by really improving that read length performance and giving us then more complete transcripts for the study of transcript information. So up to this point, I've talked about sort of like the standard transcriptomic approaches where you take the RNA, you reverse transcribe it to cDNA, and then you sequence. And adding in full-length reads from Oxford Nanopore can enhance our ability to detect things. So we've got in the middle here, right? Novel isoforms, fusions, information that we might miss using more of a fragmented short read approach. But there's also actually another method for studying information in the transcriptomic layer, and that's actually direct RNA sequencing. The direct RNA sequencing is going to give us all the stuff we see in the middle here from a normal cDNA sequencing as well. It gives us the ability to get novel isoforms, fusions, full-length reads. We have a barcoding kit, so you can sort of be dynamic with the number of samples you run. But it also adds in the ability to detect and visualize RNA modifications, and it has the benefit of being PCR-free. So you're not going to need to worry as much about PCR bias, reverse transcription bias. You're going to just take the RNA and you'll be able to sequence. And so this kit actually is a really good transition to the next part of our story because, talking about the differences in RNA levels and transcript levels, but RNA is not just expression and splicing. It's also got this chemical modification layer that adds an additional layer of gene regulation that we could potentially visualize with direct RNA sequencing. So far we've talked about RNA abundance and RNA structure, but RNA biology also is going to include that chemical modifications on the RNA themselves. And this is typically lost when we do our conversion to cDNA because we're no longer looking at the RNA, right? We're looking at the cDNA molecule. So I'll touch on this briefly from the transcriptomics perspective because direct RNA sequencing enables us to look at native RNA modifications. Now, I should note this is distinct from the DNA modification, or excuse me, DNA methylation layer. That's going to be covered next. But conceptually, it's going to reinforce that same multiomic message, right? It's that biology is not just regulated at abundance of a gene or what SNPs or fusions are present at the DNA. Biology is regulated at multiple molecular levels, and that's the reason that we need to be, or at least considering, multiomics in our complex disease research and our research in general. Because RNA modifications, like methylations, for example, can do things like in influence RNA stability, can influence localization, interactions with RNA binding proteins. And they're increasingly being linked to different human diseases, including cancer, neurological disease. They're being studied in immune biology, developmental disorders. And so transcriptomic assays that can provide more than just a readout of expression, give us the full-length transcripts and get us transcripts, get full-length transcripts and give us transcript information, but also giving us modification information can really help reveal how the RNA molecule is processed and modified as well. And this is just to show that, the epigenetic field, so this idea of studying the modifications that are on the RNA itself, really quite a growing field. We've seen quite a few publications over the years with quite a nice uptick in the past 5 years or so. And this just showing that in general, high impact papers and high impact journals are highlighting things that are talking about methylations and modifications at both the RNA and the DNA level. So just one example from the literature, quick to illustrate where RNA modifications can matter biologically. So here we're looking at RNA methylation, specifically methyl 5C of transcript RNAs, tRNAs. So mutations in the methyl 5C RNA methyltransferase NSUN2 are associated with microcephaly and other neurological abnormalities. And so NSUN2 mediated methylation of tRNAs. So what ends up happening is it protects those tRNAs from cleavage into smaller RNA fragments. When that methylation is disrupted, tRNA becomes more susceptible to cleavage. This generates more of those small RNA fragments. This can drive cellular stress, and cellular stress can result in cell death. And these modifications really they're not just chemical annotation on an RNA. They really have a functional consequence for that RNA with regard to the stability. And then you see downstream causes things like additional cell death, as we're seeing here in cells that are lacking NSUN2, higher level of dead cells following apoptotic stimuli. So UVB, for example, is an apoptosis inducer. And you can see that when you don't have that particular methyltransferase, you see a higher fraction of those cells undergoing apoptosis and dying as a result. So if we want to understand disease mechanisms at an RNA level, we can benefit not just from capturing expression, but also from RNA processing and understanding the modification status as well. So just to close, there's a lot of information available in the transcriptomic layer to help us understand disease states in biology. Oxford Nanopore actually has 2 different complementary solutions to study transcriptomics at a bulk level. For researchers focused on scalable isoform discovery, you want a lot of reads, you're doing things with transcript annotation, maybe fusions. Our cDNA PCR sequencing solution can give you that long read information and some good quality scale of the number of reads available. For researchers that really are, maybe curious about the modifications that are present in their RNA, maybe they're interested in poly A tail length, things like that, which you can get if you're not reverse transcribing your RNA to cDNA. Direct RNA sequencing is going to provide some really unique and powerful capabilities to study that transcriptomic layer, not just at the gene count, but the transcript level and also at potentially the modification level. The broader message here is that transcriptomics, as Oxford Nanopore thinks about it, really allows researchers to go just beyond that expression level. So we can move from the DNA layer, see what's there, what type of SNPs and things might be present at the DNA level, start to look at the regulation and the expression of RNAs, and then start to understand also, how these RNAs might be getting spliced, how they might be getting processed, and ultimately how they might be getting chemically modified that could result in their in sort of differences in processing and such. And when we combine these analyses together, it's just going to give us a more complete multiomic view of complex disease biology. It's really going to help us understand how the genomic variation can influence the regulatory state, can influence the RNA output, and so on. Great, thank you.